Yeah, thank you for the introduction. So yeah, I'm a PhD student in Gina Gagneur's group and Fabian Theis's group at the University of Munich. And I want to tell you a bit about Scooby, which is a model that we recently developed that is one of these big models that Cassandra just mentioned that goes from DNA sequence and tries to predict RNA and attack profiles at single cell resolution. So why are we interested in these models in the first place? Me personally, I was just interested. All of our cells, they kind of carry the same genome, or at least the healthy ones. And yet we have all of these super diverse cell types in our body. And it's really just it's the regulation of the gene expression. And this is such a tightly regulated process that orchestrates the cell differentiation. So if we understand this process, we eventually can also understand how do variants interfere with this process? At what stage do variants interfere with this process, which could in principle lead to some diseases? So our lab, but also other labs, we saw a lot of talks already yesterday, try to tackle this question by basically taking or using models that take DNA sequence as input and try to predict a phenotypic or molecular phenotype from this. And the goal here is not really, and Jacob also mentioned this nicely yesterday, the goal is not really the prediction because we measure the data, but the goal is to have a model that we can then interpret, and have these models allows us to find regulators, predict effects or predict effects of variants, and lastly also to maybe design elements that have a property that we would like them to have. And there are lots of methods that came out there. I'm just going to use Borzoi now as an example. There's been Alpha Genome coming out just recently, but they all were kind of similar, that they take a large chunk of DNA as input, and then they feed them through diverse neural networks. And these neural networks should learn a sequence embedding, which should be a richer representation of what the sequence actually contains. So it should be are there transcription factor motifs in the sequence? Am I in an exon? Am I in an intron? And then these last layers here, they just take the sequence embedding and they decode this into the respective assays. So in the case of Borzoi, they predict a tax-seq signal, DNA signal, bulk ChIP-seq, bulk RNA, and cage-seq. I just mentioned, so all of these methods, they are trained on bulk data, so they don't really capture the cell type heterogeneity, which is not really a problem, but I was interested, or we were interested in a tissue that consists of, here I'm showing you one example of a bone marrow tissue that is single cell multiome data, and we see there are a lot of different cell types in there, and there are also these transitions within these cell types. So for example, these hematopoietic stem cells that they differentiate into normoblasts or erythrocyte cells. So we're asking the question, can we use these pseudobulk trained models and bring them to a higher resolution that we can also interpret this data? Because this would allow us to find transcription factor activity for transitions, or find sequence determinants that are maybe guiding a cell fate transition. And lastly, also there might be variants that only are affecting a specific cell state and not others. And the main challenge that we actually face, because the large models are already super computational expensive, how do we scale this to millions of cells? How do we even do this? So this is how we came up with Scooby. And Scooby, I'll walk through the different aspects of what it does in a bit, but at the core, it actually takes Borzoi as its foundation model. So it takes the Borzoi model. The only thing that we add here is these dataset specific parameters. So in our head, they should give the model some capability to actually adjust to the single cell data sets in the first place, but also maybe upweight or downweight specific motifs depending on what data set you're looking at. And then the core of it is actually this, what we call the lightweight cell-specific decoder. And here we're making use of these embedding vectors. So you've seen a lot of embeddings already in the previous talks, but at the core of them, they basically measure how similar are different cells. And this gives the model a notion of what cell type or what cell state am I actually looking at. And the cell-specific decoder, it takes both these input and the sequence embedding and tries to decode this in a cell-specific manner. And how I think about it is that it gives a because these sequence embeddings, it contains information about transcription factor motifs, but they need to be decoded differently depending on if I'm in a T cell, on a B cell, or a different cell state. So it takes all of this together, and then it predicts RNA-seq coverage and attack insertion coverage at single cell level. So how does this actually look like? And first of all, I want to give you like an impression of how the actual data looks like that we're trying to train on. So we heard a lot that single cell data is very sparse, and this is also what we see. So this is an RNA-seq profile for a single cell. And when we actually look at the prediction that Scooby does, it looks like a lot less sparse, actually. So it's kind of capturing a profile across the cell. So we thought like that's probably like somewhat the expected profile of this cell. So when we look at this now in this matrix here, each line is basically one of these single cell profiles. So and these are the profiles of this one cell that I showed you and all the 100 nearest neighbors, which should be somewhat similar to this one cell. So if we aggregate this, we can think of this aggregated profile kind of as the expected profile of the cell. And then if we compare this to the Scooby prediction, we actually see that they kind of align very nicely. And this is maybe even a bit more impressive on the attack, because attack is even a lot sparser than the RNA-seq data. But even there, the Scooby kind of manages to aggregate this into like an expected profile. When we also wanted to evaluate this a bit more quantitatively. So as our like bottom baseline, we were like asking how well could we actually impute or correlate these very large profiles when we look at the corresponding pseudobulk profile, which is the box I'm showing you here. And the Scooby predictions on the single cell profile, they are definitely better than this just using the pseudobulk baseline. And as an upper limit, we thought that the profile of the 100 nearest neighbors, the single cells, we thought of them as like an upper baseline. But as I showed you previously, Scooby, the profile looks more like this expected profile. So we also correlated them with the profile of the 100 nearest neighbors of every single cell. And there we actually see like a very high correlation with the profile that we predict. And the same is also true for attack. So then a bit more metrics, we went into how well do we actually predict gene expression. So as I mentioned, we are predicting the read count. So to get a gene expression count, we then just aggregated this profile across all exons to get a gene count per cell. And then we averaged all cells into pseudobulks to compare the metrics. And you can think of two different ways here to look at the metrics. One of them is for each cell type, we look at how well we predict the gene expression profile, which is what I'm showing you here. So we get really high Pearson correlation for this. The harder task is actually if for one gene we would like to differentiate between cell types or cells, where we also do decently well if you compare this to like Borzoi, for example, but for all of the single cell of all of the sequence-based models, there's definitely still room for improvement here. So now I want to show you two use cases that you could use this model for. One of them is this inferring transcription factor activity for like cell states. And for this, we kind of came up with a method of how to interrogate the models for like a transcription factor activity. And what we did is that we scanned we scanned the sequences for transcription factor motif instances, and then we just randomly replaced them with other sequences, and we recorded the difference on the gene expression. And you can do the same for attack. We also show this in the preprint. So the difference between the predictions with the motif and without the motif is basically an indicator of how important this motif is for a cell. So to give you an example of how this looks like, this is now the early B cell factor, which is a master regulator for B cells. And Scooby actually predicts, so this is the transcription factor motif activity, and predicts a very high activity here in the B cells. But of course, we can also evaluate this a bit more quantitatively. So what we did here is also what Cassandra mentioned. We correlated the motif activities, not the transcription factor activities, with the TF expression. So each dot here is one of these correlations for Scooby, and we compared this to a method Cromwell, which only uses accessibility data. And we see that we better correlate than Cromba in this task. But interestingly, because you could think, oh yeah, but you're using ATAC, we also trained a model on just the RNA, and even on with a model that has only seen the RNA and tried to infer the motive activity from there. We also see a better correlation here. So I talked about cell transitions. So I want to give you one example where we actually tried to look into this. So this is now a different data set. This is a heart organoid data set, and they measured at different time points, like heart development within these organoids. And in this publication, they were very interested in this population here because it has like a dual fate. It can differentiate both into the epicardial fate and the cardiomyocyte fate. And in this publication back then, they identified different cell populations which have different transition probabilities to either the epicardial fate or the cardiomyocyte fate. And we were asking, can we identify transcription factor motives together? And I think there are two interesting things in this plot. First of all, I thought it was cool that Scooby can identify these differences even within its cell type. And also these transcription factors, they were kind of, especially GATA4 was kind of known to also be driving this cardiomyocyte fate. And of course, you can do this more systematically and scan like different transcription factors for their fate probability. And lastly, I want to go a bit more into the variant effect prediction, which has been like a kind of, or not debated, but like everyone wants to do this. And here we were asking, okay, are there variants that are affecting just one cell type and maybe not the other, because it might be destroying a transcription factor motive that is only relevant in a specific cell type. And to benchmark this, we used two different data sets. One of them is the 1K1K data set, which they measured EQTLs for 14 different cell populations. So you somewhat have a cell type specific EQTL effect. And the other one is GTEx, so this is a bulk measurement, but we just included this to have to evaluate how much do we actually lose maybe on the bulk EQTL data. So for each of these EQTL variant pairs, we basically compute a variant effect score by replacing the comparing the prediction without the variant to the prediction with the variant and using this as our variant effect score. And then we correlate these predicted effect scores to the actual observed scores. And we compare, here in this plot, we compare these correlations of Scooby against Bullseye. And first of all, we can look at this blue dot here, which is this bulky QTLs that I mentioned. So we see that we don't really lose so much compared to Bullseye, which has also seen this GTEx data set when moving to single cell data. But on the cell type level EQTLs, we actually improve on all of the cell types that in the data set. And so this is all nice, but we wanted to look a bit more into which variants do we actually get right, and is there maybe some room for improvement? So we looked specifically into these GTEx EQTLs. And one thing that we observed, which has also been reported already yesterday, is that for a lot of variants, we do predict an effect, but actually for many more, like the sequence-based models, they just don't predict an effect. Another thing that we wanted to look into is like, for how many variants do we actually predict the correct sign? So this is why we have these percentages here in the different quartiles. And we, like, for most of the variants, we see a kind of the good sign. But one thing that we were looking at, what if we actually filter for variants where we predict a significant effect or where we predict some effect? And if you filter for those, we actually, first of all, we see that the correlation goes up, but also we see that if we filter, then actually most of these variants that we predict the correct sign for them. And we saw the same for Bullseye. So yeah, there's still some debate on what this means. But another thing that you could also look at, and this has also been investigated, is how well do we predict variants that are further away from the transcription start site? So there's been some work that showed that the closer you are to your transcription start site, the better you are. So here I'm showing you again the the sign concordance of our predictions as a function of the distance to the TSS. So we also see, we had hoped first, like maybe if we go single cell, we don't see this decay, but we also see this decay. But another message that I want to give you is actually if we filter again to the ones where we do see an effect, we don't really see a decay. So the take-home message here for us was like, you can kind of trust the predictions where Scooby and also other sequence models predict an effect, but there are also a lot of them where they just don't predict an effect. So in conclusion, I presented you Scooby, which is a model that works on Borzoi, and it has this lightweight decoder to predict at single cell resolution, and we kind of accurately model accessibility and expression profiles. We can interpret these models for cell state specific sequence determinants. And I mean, I showed all the work on Borzoi now, but in principle, you could think of applying this to any sequence-based model because it's kind of like, just on top of the sequence embeddings. And with this, I would like to especially thank Johannes Singer, who is a co-first author also on this paper, and my 2 labs, of course, and all the co-authors on the paper. And if you're interested, I mean, I just showed a very brief summary of what we did, and there's a lot more in the preprint. Thank you.
Thank you very much for a fantastic talk. I'm sure that there are questions. We have a bit of time. Yeah.
Thanks so much for your talk. I was wondering, so eQTL datasets are not fully saturated in their power. Are you going to be able to use this method to help to prioritize variants so that eQTL discovery can be improved?
I mean, we didn't do it in this way because Borzoi kind of tried to already benchmark this a bit. How well do we prioritize like a fine mapping of variants? In our opinion, like maybe it's more reasonable that you combine them, so you still have the power of the eQTL data sets, but you could increase maybe for variants where you're not so sure. You can like increase it with the Borzoi results, and I think David Kelly just recently had a preprint actually where he tried to combine both of that together. Yeah.
Online question next.
Yes, from Vitaly Klashchevnikov. How different can pseudo-bulk models be to the single cell data for this approach to still work?
How different?
How different?
Pseudo-bulk data be?
Pseudo-bulk models. Bulk models.
You mean like how? I guess.
I don't mean. I mean.
I don't exactly understand his question, but I mean, there's Decima, for example, which is a model that tries to predict pseudo-bugged gene expression data. We haven't really benchmarked it yet because we were kind of came out at the same time, but like in my head, they are not really exclusive, like having a pseudo-bugged single cell data, pseudo-bugged expression data and having Scooby, because in principle, you could think of having a pseudo-bugged model which has a better representation maybe than Borzoi, and you still use Scooby to resolve the single cell.
How different can Borzoi?
Yeah. You mean how different our measures? I mean, we tried a model originally where because this was like we didn't believe ourselves that we could predict the single cell profile. So originally we went first pseudobug, then we went 100 nearest neighbors. And then at some point we was okay, let's just try single cell. So actually the models that were trained on single cell versus 100 nearest neighbors, they performed actually completely similar, so all the same. So that's why we decided in the end to go with single cell, because then you don't have to have this, oh, we only take 100 nearest neighbors, or we take this weird aggregation. Yeah.
Here.
Yeah.
Could you backprop all the way up to the inputs of the embedding model to calculate feature contributions in the ATAC-seq or RNA-seq space?
Yeah, that's like a different way that people also do these motif effects, where you like try to do like gradient types input, for example, to get feature features that are predictive of your output.
Good. Let's thanks Laura again.